Okay. All right. Well, I'm going to go ahead and get started. And as everybody kind of comes in, they can get logged on. Well, as most of you know, my name is Casey Craig, and I am the Executive Director for the Cute Syndrome Foundation. And I'm also mother to Miss Stella, which is an SENAA warrior. She turns 9 this month. I can't hardly believe it. And this is just really, really exciting for me to be in this role as executive director and be able to serve all of our families and know kind of, where you're coming from. We're all on a different journey, but we all have our special kiddos that we love so dearly. And so today is actually our very first episode of the Family and Researcher Roundtable. And today I have 3 amazing guests from Praxis. And so I'm sure most of you, whenever the press release came out about the phase 2 results from EMBOLD study, frantically went and read all the things. And you probably were on our deep dive day as well. What was that, 2 weeks ago now? Time's flying by. But if you're like me, especially before I was in this position, you're reading all this and you're like, well, this sounds exciting, but what does that mean? And so what we wanted to do today was bring on Praxis to really talk about what did the phase 2 results mean? What does that mean for our community moving forward? And then also phase 3, what does that look like? How do I enroll? Are the exclusion criteria the same as last time? So we have lots of things to talk about today. So I do want to introduce really quickly. So Julia, I know you're not going to, talk much today, but Julia Chase is our patient advocacy liaison, if you will, and she's a joy to chat with every 2 weeks. And so we're happy to have you here, Julia. And then we have Brian Spar, our Chief of Staff of Praxis. And so he's going to be jumping in throughout today as well. And then I'm going to hand it over to Brian Pfister. He's the VP of Medical Affairs. And Brian, you have such an amazing way of taking the high level science and all the details and bringing it down to a level for our families to really understand and be able to educate themselves and ask really good questions. So we're really excited to have you on today. And so as you're sharing your slides here, I will just kind of mention to the families that we actually have 2 areas that you guys can ask questions today. So I know sometimes the Q&A can be a little wonky, and so you can either ask a question in the Q&A at the bottom of your screen or in the chat section. And I will keep an eye on that as we're going through. And so if you have a question that pertains to the slide or the information we're talking about, then I'll bring it up then. If not, we will have some time at the end for some questions and answers. Okay, well, Brian, I will hand it over to you.
Excellent. Thank you, Casey. As a researcher, as a VP of Medical Affairs, and as a father of a son with epilepsy, these things are both exciting to me, as well as then, it's very unique opportunity to be able to talk to families. So I appreciate that more than you guys can realize, to help you understand and get questions answered, and hopefully help you on your journey along the way. So thank you for letting me be here today. And what we want to talk about, as you mentioned, is the top line data, obviously, but we do want to highlight the process. Like getting data takes a while, and it can be very frustrating. I just wanted to highlight, what does the clinical development process look like, and what are the steps along the way? So we'll talk about, again, our commitment to epilepsy. I always kind of joke it's in our DNA. Really, the heart of what we do is we're trying to make lives better, and do it as fast as we possibly can. But again, I want to show you that drug development roadmap, what it looks like to get a drug from concept all the way then to market. Talk about our data, and then show what's next, and really how families can get involved to make a difference. So as I mentioned, I have a son with epilepsy. So the reason why my why do I get up every day and why do I do what I do, actually relates to him as well as your families. To be honest, our goal is to make lives better, and really to help patients by delivering life altering treatments faster and more effectively than it has ever been done before, and do it again and again. Right. So that's a mouthful, but honestly, that's our commitment to epilepsy is strong, We really love to be able to help families. That's why today is so exciting because again, sharing top line data that you're somewhat familiar with, but getting into more nuts and bolts, it helps us to deliver on this mission, and it helps us to do this in a way that people haven't really done before. So we'll highlight that through the course of discussion today. So here's the brief review of drug development. So you may have seen animations like this or slides like this before. For us, when it's about families, when it's about bringing medicines to market and really making a difference, time is always of the essence, right? So for us, the urgency is there. We want to do better. We want to do better today, but we understand there is a process that clinical trials typically have to follow. This is outlined below, so you can see, when you look at it from left to right, there's the early discovery, preclinical studies. Then we go into phase one studies, phase two, phase three, and ultimately then to approval. So I know we've heard those terms used a lot: phase one, two, three, those type of deals. I just wanted to kind of, break it down into a little bit more detail for you of what that looks like. So one thing to note is, while this is a traditional quote unquote development process, not all genetic conditions and not all genetic treatments follow the same pathway. So there can be variations along the way, and there can be opportunities for us to accelerate wherever possible. So just wanted to highlight that. And kind of the underlying component or the red thread through all of this is there's always the regulatory agencies that we're in contact with to let them know of what our plans are, what this looks like, and share our data along the way as well.
I wanted to point out too, just looking at how old SCNAA is, from the discovery to where we are today, that seems remarkable speed to me. Would you say that that's been, what you've seen as well on your end?
Yeah, it's been it's kind of the Praxis way. We want to go as fast as possible. So seeing this and seeing how rapidly it's evolved and how rapidly we can get on target treatments, and I'll kind of talk about that in a minute too. But to get drugs that are effective and safe in that space in the time that we're
Mhm.
doing it, I think is faster than I've seen before. So it's really, I think, a testament to the mentality of the families to be able to participate and to be in this, because without the family participation, we don't have top line data, right? So I
Right.
don't want to put too fine a point on it, but when families get involved and they really are part of the process, we can move much quicker together. So again, kudos to the families, honestly, for being part of this and
Absolutely.
helping us to get there. But when we look at, and I think it's a good point. And when we look at what we're doing, what is the goal of development? We're trying to find drugs, medicines that work, that really have the activity where we want them to be. So this happens in early discovery. So this is kind of the like come up with an idea for a drug, look at what it is. Could be a protein, could be a receptor. There's a whole host of different targets, and a lot of times there's multiple agents that are being looked at once. So we say, here's 5 or 6 drugs. Are they going to work in the space? And so we don't always know immediately. Theoretically, we believe that they would work, but we don't always know immediately. So we want to test them, in early discovery to look at it. Can we find what seems to be the best of the bunch to move forward into the next phase of development? And those next phases tend to be the preclinical studies. So these are in cells, small animal models, even computer simulations, because what we want to do and what we want to make sure is each step along the way that we identify drugs that are going to be what we believe to be efficacious, but we want them to be safe, right? So safety is always top of mind for us. And so these preclinical models will help us to identify early if there's a problem, if there's something that happens, if there's an issue. So each step is gating for us, and looking at the data to determine what the next steps are, because we want to be confident that the drugs are going to be effective and safe, but we don't want to sacrifice any quality. And then we get into phase one. So phase one, after we have identified the molecule, tested it, believe it to be safe, then we'll look at it in healthy volunteers. So phase one are going to be subjects who volunteer their time to take the medication. They typically don't have the condition, so it would be something that would, in this case, not help their seizures because they're not having any. but we can understand what's the tolerability, how once it's in a human, how tolerable is it? We can learn the safety, and we can look at any projected side effects to really understand where the dose should be. So, and I'll show you a little bit of phase one data later, where a lot of these patients end up taking doses much higher than what we'd expect to use, in a patient. So it's important to see the rigor that these things go through along the way to make sure that safety is never compromised. So when we get to the proof of concept, after we make it through the phase one and everything looks good, then we'll go into proof of concept. So this is going to be where we look at patients, and typically the phase two is what this is identified as. It's going to be smaller than your phase three trials, but what it is we want to establish that the drug is actually working in the fashion, in the way that we want it to, in the patients who are affected with the condition. So this is going to be in the target condition. This is going to be a small group of patients, to look for that safety signal and look for potential efficacy as well. Once we see that, once the proof of concept is established, then we'll move into the phase three, and we'll have confidence going into phase three that it should work in those patients, and it should be safe based upon what we learned from the other stages of development. So really, when you look at it, there's not a lot of phases you can skip, but there's ways that we can accelerate that, even though we can't skip a phase, if you will. And ultimately, when we have the full data set and we've gone through the phases of development, and we'll submit that to the FDA, the regulatory agencies will look at this. They'll make sure that they agree with our conclusions, that we've established the safety, efficacy, and tolerability profile, that would make the drug approvable and used in multitude of patients. So that's the basic overview of drug development. Each step is important. Each step is again, a bit of information and evidence, if you will, that allows us to make decisions, allows us to move into the next phases. So I want to show you what we've done here with regard to our relutrigine, which is going to be used in SCN8A patients. But I want to show you the process that we took and things that we've done to enhance the drug development process and make it much better for families. So when you look at this is just kind of highlighting, up into the phase one component. So we did go through the early discovery. We looked at a multitude of candidates. We picked relutrigine because it did have the qualities we were looking for. We believed it would act in a particular way, and we tested it in the preclinical studies. We found it to be safe in the models that we used. We also looked for signals of efficacy in the models that we used as well. So we did see those. So what is exciting about that is the lights are starting to turn green, which allows us to keep moving forward. And so then you go into, again, phase one with the healthy volunteers. What's interesting about relutrigine is it's called a functional state modulator. And I'll show you what that means on the next slide. But what we're excited about is we really believe it has the potential to be the first disease-modifying treatment for SCN8A DEEs. Because a lot of times what we've seen in the past with drug development is there's trade-offs that have to be made. There's certain drugs that may work really well, but I don't really like them a whole lot. And I have my short list. I won't bore you with those details today, but definitely there tends to be a trade-off historically, what we've seen with development of anti-epileptic or anti-seizure medications. So this is the way that the drug works. So this is our theoretical orientation. We've designed the molecule to act in a particular way. So for relutrigine, what's really unique and different about this as compared to other sodium channel drugs that we've seen, most sodium channel drugs will block everything. So if you take these channels that are normal functioning, open, closing, inactivating, they'll block everything. And so what that means is that's where you start to get into your trade-off. You start getting like, okay, we can have seizure control, but we're going to lose certain aspects, I think, of the child's personality or the patient's personality in general. So what our goal is to design a drug that actually would work in this pathological mutation area. So working where it needed to, but leaving these channels untouched. So what that would mean is if they're open, closing, and inactivating normally, it's not going to block those, so they'll be functioning as appropriate. We'll just address the hyperexcitability, and by that, this is what typically is causing the issues with the seizures, frequency, duration, relatively low threshold to trigger seizures. And what we're focusing here is really this enhanced persistent current. So the persistent current, if you think about it like a faucet that just keeps drip, 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 and you had a bucket that fills up under it. Once the bucket's full, it'll tip over. So you have this drip, drip, drip until you trigger a seizure. So what we need to do is most of the drugs that are available don't really address persistent current. So this is a first in class. This is the first drug that's really going to address the needs of the patient and focused on this hyperexcitability where the particular SCN8A mutation happens in this channel. So what this means is we've designed it also with some really, I think, important aspects. QD dosing, which means once daily. So you've seen some other medications that are, like my house, we're on twice daily. I don't really love that, but it is what it is at the moment. So drugs that are QD dosing means just once a day. It doesn't require titration. So that is realistically one of the most important pieces that we see, because that means we don't have to wait 6, 8, 12 weeks before we get to a level that might have benefit. We can start immediately at levels that we know to be have benefit for the patients. This, I think, becomes a game changer. I think this molecule, if I were to put my dad hat on, this is a game changer, right? This is something that is holistically different than what we've seen, and it's been designed to be different. So it's not just serendipitously different. It is, intentfully designed to be different, and really intentfully designed to help the SCN8A community, because we've seen, and as we've studied and learned about it, we've really seen that there is a huge opportunity with the way this drug works to drive the outcomes for the patients.
I love that slide, Brian. And I think, and again, just to kind of bring it back to the families as well, is the majority of us, I would say, are on a large number of sodium channel blockers. And I'm just envisioning, I'll use Stella as an example, and she continues to see, so we keep increasing these sodium channel blockers. And what they're doing is just dampening, they're working on all of them, even the ones that are working well. And so we might get that seizure control, but then that's where we're, getting the major side effects. And then we usually end up having to, pull back because of the side effects. So I love that it's working on the mutated area, and leaving the other things that are working well alone, right? But then also the no titration. I mean, I just really want to hit home for that for families. Wow. I mean, that's a really big deal. Most of the medications we have to titrate, or, you're in a crisis mode, and that's why you need to make a medication change. And it's just infuriating that you have to wait, sometimes that 3 months or longer to get to that dose. So that's really, really exciting.
Yeah, no, I agree with you. The titration one is the one that gives me goosebumps because it is so
Yeah.
different, and it means it has such a meaningful impact to families. If you can start it today and you're going to have some potential benefit and it immediately kicks in, that's a huge, huge benefit. And you don't have to then worry necessarily about the risks associated with other sodium channel drugs, because again, this acts differently and operates differently. And structurally, it's not like the other sodium channel drugs too, which contributes to the titration schedule.
Yeah. Awesome.
Yeah, and so what this is an important slide for me. There's a lot of things on here. I won't go through each and everything because there's a lot of pieces. But when we get into the how do we know it's acting differently? What evidence is there to support that this drug is not acting like what we've seen before? So when we look at the healthy volunteer data, the summary data we have here, 130 healthy volunteers have taken this product. And the 130 healthy volunteers that have taken the product have taken it at higher doses than what we would expect to use in the clinic, or higher doses than what we would expect to treat the children with. And this little abbreviation, MTD, means maximum tolerated dose. So typically, if you give somebody who doesn't have epilepsy an anti-seizure medication that blocks sodium channels, or that would affect sodium channels, they would get very sick, right? They would get dizzy, they would get confused, they would have ataxia, not being able to walk properly. They would feel kind of drunk, if you will. So, and then they would say, okay, no more, I tap out, I can't take this anymore. And that would be your MTD. That would be the maximum tolerated dose before patients would drop out. So I don't think I've ever seen this in a drug like this before, where you didn't reach a maximum tolerated dose. That means the patients kept going. And even when you got to where you thought you would need to be to control seizures, they could even go higher without having issues and tolerability. So this is how I know in my researcher hat that this is different than what we've seen before. And this is really, like, I think, an important piece when we look at it, that it is has been tested in a lot of patients. And of the 130, not one has reached its maximum tolerated dose, which is really important. So that allows us, once we have these data points and this evidence, this allows us to move very confidently and swiftly into the next phase of development. And so that's then the EMBOLD clinical study, the phase two. So this is children with SCN2A and 8A. And again, this builds on the chain here. If you look, we're adding more pieces to the puzzle as we go along here. So before I show you what the results are, I want to tell you the how. Like, how did we do this? Because this is also something I haven't seen. So there's a lot of what has Brian never seen before. So this is something I haven't seen before, but I'm happy to have been part of. Like, really, really happy to have been part of this, because this represents in a lot of ways the voice of the patient, right? So we heard traveling to a site is not, if not impossible, improbable. So we're like, okay, we believe so strongly in what this mechanism of this drug is and the potential benefit to have impact on these families, that I've never seen a company who says, let's do something no one else has done in this space. And so what we had done as a team is come up with a way that everything could be done at home. So there's no playbook. I go back to the phase of development. There's typically like a playbook and there's ways to do it. There wasn't one for this. And so, I mean, the number of hours that people on our team spent sorting this out, and the number of weekends and the number of holidays that were spent to make this happen is huge. But it's so important to us that we be able to create an opportunity for families who, like I've said in the past, who couldn't have been part of a clinical trial, only for the fact that they had a much more severe presentation of the condition, right? These are kids who couldn't have traveled to a site, but now they're able to be seen at home, right? And so we want to continue this because it's very, very important to the families, to be able to create an opportunity to do everything at home. You know, nurses and physicians come to your house to assess your child. It creates a nice safety piece as well, right? So you're not having to worry about traveling and not having to worry about, sleep deprivation at our house becomes a problem with seizures every now and again. So we're not having this issue, which allows us to really better study the drug, to be honest. But in the same vein, it helps us to get families who want to participate who just could not historically. So again, I'm very proud of this as a company, very proud of this as a team. It was a lot of work, but well worth it. The way the study was designed, and this slide gives you kind of more of the schematics, because there's another really cool aspect of this trial that sometimes isn't always fully understood. So we did randomize to two treatment groups. So we had one, which was relutrigine for four dosing periods of 4 weeks. And then we had placebo. But what we wanted to do is we wanted to limit the amount of placebo exposure, right? So what this means is, instead of having 16 weeks of treatment and 16 weeks of placebo, we said, if we do it the right way, we could probably limit the exposure to placebo, still get the answer that we're looking for, and be able to continue the trial into the next phases. So in this trial, there was only one period of 4 weeks in which the patients were on placebo. So that means 75% of the time, everybody was on drug, which I haven't seen before, but I highly, highly applaud, the effort that went into making that happen. And then we were looking at the key endpoints. Obviously, these are very common, but we're looking at seizure freedom rates. Like, do we have seizure freedom? Changing in baseline from seizures to after treatment. Looking at adverse events, obviously safety, as I highlighted before, so important. But then we also looked at some other things down here. This one I think is really important too, because this is the clinical global impression of improvement and severity. So we look at baseline, how severe is the patient, and we look at the end of treatment and say, did anything change, right? And so this is going to get into some of the qualitative and functional changes, which we've heard repeatedly are very important. So this gets into that trade-off component you mentioned, right? And this is again, I guess I have a lot of goosebump moments, but this is also a really cool one too, and I'll show you that data here in a minute. But it's really important to be creative when we're trying to go fast, but we need a lot of data. We need a lot of pieces to make decisions so we can do this the right way. So again, kudos to the Praxis team for the way this was designed and executed. So here are the demographics and baseline characteristics. So we look at the mean age in the placebo group versus total. So you've got two columns. One is going to be the placebo group, and one's going to be the then total when everybody's combined together. So we look at the mean age, about 6 years old, so we can get an idea. Nearly evenly split with the 8A and 2A patients. Uh, Gender in split was nearly even, slightly skewed towards the male side in this one. We looked at age of onset, so looking at early onset, if you will. So these are going to be, you can see the data on when the seizures first were occurring. Most of them were happening in the first 3 months of life, again, which is consistent with what we would see with a gain of function situation where you have this hyperexcitability in the channels as well. We look at the baseline anti-seizure medications being used. And again, these kids have been on a number of meds. Like it isn't as though this is the first time they're seeing this and giving go. So the families had been through a lot of seizure medication changes and kind of trying to dial in the dose and still not quite where they wanted to be. When you look at the baseline motor seizures in the area before being treated, you had about on average about 50 to 53 seizures. So it's quite high seizure burden. And then when you look at the severity, these kids were marked as pretty severe in this situation. So gives you an idea of who we studied. And so then we look at safety. So whenever we talk about efficacy, we want to talk about safety as well to make sure that we create a safe product first. So when we look at the placebo, you can look at these columns and just bear in mind placebo is only 4 weeks. So for majority of the time, the patients were not on placebo. But when you look at the comparison to placebo versus treated, it's a very, very favorable safety profile. In a lot of ways, nothing stood out as being a major concern. We didn't see any drug-related SAEs. We didn't see any discontinuations due to the drug. They didn't come off of the relutrigine, and most of the adverse events were mild to moderate and no major issues that had popped up here. So this is encouraging because it allows us to, again, feel comfortable with what we saw from an efficacy standpoint. So this is the efficacy. And I want to orient you to this number here. So when we look at the total motor seizures at baseline, there are a lot of these. So in 15 kids, there's 1778. That's a lot of seizures. So to be able to reduce that by 46%, and this is placebo adjustments in there, to be able to reduce that uh, frequency by 46%, I think is a really, really important figure. Because this is, again, we haven't seen things like this before. And so that goes back to building on, the MOA is different, the idea, the study is different. All these things are different. And ultimately, then we can, you know, be assured in this that the drug is working. And so what's really, really exciting to me is in the patients, in 5 of the 15 patients, they had seizure freedom. And so this is like, while it's always what your treatment goal is, historically, I don't think that's always been what people actually thought was realistic, right? So the game changer component, now our expectations, at least my expectations in this space, change. Now this becomes a very reasonable and likely target, with these type of medications. So I couldn't be more proud of this one because I know what it's like at home to not have seizures, and I know like that first time that you're not having seizures, it's like really, it's a major event. It's a major milestone in a lot of ways. And what was really nice to see too is up to 200 days, and it's longer since when the data were looked at, the patients were still seizure free. So it's not like, oh, we just had a blip and maybe had a couple of good days or had a good month. This is like 6 months, over 6 months worth of seizure freedom for these families. So I'm incredibly encouraged by these data, like beyond encouraged. And as you mentioned before, and again, the trade-off. So what, to see results like this, was there a trade-off involved? And I would say no. I would say in this situation, we didn't see a trade-off. We actually saw we looked at the CGI-I, so clinical global impression of improvement. What we saw is behavior got better. We saw communication improved as well. So you can see the dark bar is the clinician, the light blue bar is the caregiver. So what's interesting here is both the caregiver and the clinician recognize this benefit, recognize this change. And so this is where we believe relutrigine, it can lead to disease modification because you're not making that trade-off. You're not making that I can get seizure freedom, but I'm going to lose function. And here we actually see over time that the seizure severity and intensity improved. So less intense seizures, less severe seizures, which also gives a bit of peace of mind. And this one I think is always the exciting, right? So the alertness, like okay, we controlled seizures and we got to seizure freedom, but did that come at a cost? And actually, no, we saw an improvement, a very, very robust improvement, as rated by both clinician and caregiver, that the patients were more alert, they're more aware, on top of having better seizure control, on top of having non-titrated drug, on top of having something holistically new. This, again, to me this changes what our expectations are for treatments.
I was just going to
So when
jump
you
in
look,
really quick,
yep,
quickly. Sorry, Brian, is when you can actually go on to the next slide and that 46% seizure reduction. I know the conversations that we've had with Stella's epileptologist, and a lot of times those conversations are, well, if we can get a 10% improvement, that's a win for us, and then I'm sure a lot of families at home are having those similar conversations. So to think that 46% is a possible outcome is really, really exciting.
yeah, and building upon that idea too, and this kind of highlights here, it actually jumped to 75%. So the longer the patients were on it, so this is 46% in a 16-week period, right? So that's real.
Mm-hmm.
Like going back to the titration, you mentioned it could be 3 months before you get to where that dose could be. So in that time that it takes typical drugs to titrate to a safe level in a safe manner, you already got 46% seizure reduction. And the longer then they stay on it, then you see like with that severity and intensity decreasing over time, ultimately you get then to 75% long-term seizure reduction. Those things are just, we have not seen. We just
Mm-hmm.
simply have not seen that with new molecules that we've developed in the past in other lives, other companies. We just haven't seen it. So this I think is important. And to be well tolerated in a population that has multiple, ASMs on board is really important. The seizure freedom, for longer than 28 days is important. And looking at the functional piece again, as the father hat, I'm like, oh, this is really exciting to me because you don't want to see if you didn't have to have a trade-off, I wouldn't take a trade-off, right? And so I think we're moving
Mm-hmm.
that way. And this data gives us an extreme amount of confidence moving into the expanded registration cohort, which I'll show you here in a minute, because this is kind of what do we do now? Like this is great. We're excited, Brian. What do we do? How do I get involved? And
Mm-hmm.
that's really where this next phase comes in is we can move this 2-3 forward, right? So we have the phase 2 results, which I showed you. Now we can move into this registrational piece, and we've done it in a way that is faster than what I've seen before, because typically there's like a really big pause in between a 2 and a 3 to revamp things. But honestly, we roll from 2 right into 3. And so that's where we are with this next phase is we're enrolling currently, and there's the QR code at the top. If families want to get involved, scan the QR code. We can make sure that they have it. They can meet with a nurse navigator to talk through any of the things and any of the questions that they might have related to it. But we're in the second half of 24 where we are now, we're doing this registration cohort. And again, you can see the design. So looking for an N of 80. So it's a relatively large number. The same trial design I showed you before. So they'll have that same type of opportunity. And then when we look at the patients who are going to be included, we 2A and 8, or yeah, so it'll be 2A and 8A, but for your families, it'll be the 8A DE diagnosis, 2 to 18 years of age, and has at least 8 countable motor seizures in the 4 weeks prior to screening and then baseline. Now, the one that's really exciting, and this is how the evidence builds on itself, is what we saw with the safety profile from the first that I showed you in the phase 2 data. Now we can be on 2 sodium channel blockers plus this. because before it was one sodium channel blocker plus this. So this opens it up, and I'm excited about this because it gives us, more families an opportunity to participate, to drive, better treatments, helping, everyone to live better in a lot of ways. So I'm very excited about this, that we did, understand the safety is there to allow for two sodium channel blockers.
Yeah, I think that's going to be huge for our community because, as you guys saw people coming in through phase two being interested in the study, I would say in the majority, if they didn't qualify, it was because of the sodium channel blocker restriction on there. But now that, we have the data showing there's not a safety concern there, I'm really excited for those families. So even, I'll just kind of plug in here for the families. If you talked to that nurse navigator when you were interested in phase two, and maybe you didn't qualify at that point in time, definitely, if it's still on your radar and this is still something you're thinking about, we want to keep pushing you to nurse navigator. I know we'll talk about that a little bit more in a bit, but it doesn't mean that you won't qualify for a phase three.
Yeah, that's a great point. It's 100%. So ultimately, when we think about this, relutrigine is really first line, best in class treatment for DEEs right now. This is what we're seeing with the data. When you look at the qualities of the medicine, it's well tolerated in the patients. It's, you don't have the trade-off, so you're not getting tolerability with less efficacy. You're getting, wonderful efficacy, with seizure control, seizure freedom, without a major side effect profile or issue. You're getting potential functional benefit. There's no need for titration. So you can start, and it's once daily. And I think the convenience, and I think this is, important in a lot of people's lives. Once daily dosing, it's oral, but J-tube and G-tube friendly. So this has been designed to be administered that way. It's also keto diet friendly. So I think that really, brings to heart what we feel to be what the families have told us, what we understand about what is needed, and how can we include this into our development programs. So again, I couldn't be happier with the data. I couldn't be more proud of the families that stepped up and were part of the phase two. Like my heart goes out to you, and just thank you for being part of this and being on this journey with us, and we're on it with you as well. So
The convenience section of this, I think, is again just to hit this home. I remember coming up with a survey and talking to parents of like, okay, how do you give your child medication so we can make this as most convenient for you? So this was another way that Praxis came to us and said, we need some input here. And we got that input from families. We talked about how they give it orally and how they give it through a J-tube or a G-tube, and the keto diet, we know how important that is for it to be keto friendly. And so again, thank you to Praxis for interested in this information, getting the information, and then listening to us and giving us something that is convenient for our families.
yeah, thank you so much. I appreciate it.
Yeah. I had a question pop up, and so I thought I would address or ask you, Brian, really quick. Parent wanted to know why 2 years old? Why is that the minimum? Is there a reason why that you can't give it to younger kiddos?
Yeah, so I'll give an answer. I don't know if Brian Spar wants to also kind of comment because he's been a major architect of this program as well. I know that was, when we had got to the regulatory agencies, that this is what they're giving us the clearance to look into, at the 2 to 18 years of age. But I don't know, Brian, if you wanted to give more color on that.
Yeah, I think it basically comes down to regulatory feedback and just iterating with them on the next steps. And now that we have patient data, we'll be in a position to go to them when we talk about the next beyond this phase or to expand the current trial. What else do they need beyond the human data we generated and any historical toxicology data that gives them the confidence that we could go to less than 2. So the goal is definitely to go down to newborn. It's just a matter of sequencing that in to meet the regulatory hurdles, both in the
And that
EU
seems to
and the
be
US.
Yeah, and that seems to be a pretty on-trend thing. As I was at the pipeline conference a couple of weeks ago, that 2 to 18 is pretty common to have that 2 years old label on there as far as starting, with clinical trials. So Yeah, it does seem to be on trend.
Yeah, there's a regulatory
Hmm.
framework you need to work within. And ultimately for safety purposes, right? We want to make sure we have the right dose for that age, for a developing child that might be different than 16-year-old, let's say.
Exactly. Yes. Okay. All right. So we have a couple more questions. Do you want me to jump in now, or do you have any, do you want to jump back into any other information, Brian?
Yeah, I'm going to stop sharing my slides so we can definitely get to the panel discussion. So, but thank you.
Awesome. Yeah, thank you for all of that information. It was really interesting to see, and very important to see those phases. I know that some parents that, were a little hesitant for phase 2 to participate, and some of that was probably a safety concern. This is the first time it's going into a 8A or 2A child. But now, we can see that it's pretty well tolerated. Again, it's a lower number. It's not like we've given it to 100 8A kids, but that all of it is looking very well as far as being highly tolerated. And then there's also that fear too of, what do I have right now? Is that the best I'm going to get? And am I willing to risk, having those, the alertness go down? Or, am I willing to risk being on a placebo for 16 weeks? So knowing that the placebo, that you're going to be on it in a very short amount of time, and the data right now is showing that there isn't much of a trade-off. And we would hope that, that continues to trend in that direction during phase 3. But very, very exciting.
Totally agree, and I
Let's,
think that was definitely the importance of helping families to see the data, so they can make that decision and say, yeah, this is something I'd like to do, or because
mhm,
again, in the early stages, you don't have the evidence, and so now the evidence is there, and I agree with you.
yeah. So with phase 3, so when we're thinking about a parent, maybe they're at home going, okay, I've heard about this, I've heard about the good results, and we're not in I don't like the place we're in, and I would like to look forward, what's our next option? And so let's kind of talk about how they would go about doing that. Obviously, having conversations with their neurologist of, where they're at, where are they on more than 2 sodium channel blockers, what does that look like? And then also let's touch a little bit on the nurse navigator. And so the families can go to the EMBOLD website, which whenever I post this in the group, I'll also make sure that link is there. And so when they talk to the nurse navigator, what can they kind of anticipate when they call them?
Yeah, so I think that the nurse navigator is going to talk through like some of the inclusion criteria that I had shown you. They'll ask questions because what we've seen sometimes families say, my son or daughter is on this medication, do they qualify? So you can kind of look at if
Mm-hmm.
there's something excluded in the history, or if you can talk through seizure frequency and type and those type of things. So the nurse navigator is someone who's not a Praxis employee, so completely confidential with the discussions, with the family. But it's there as a service for them to be able to ask questions, because when you think about a clinical trial, there's more questions than answers typically, but the nurse navigator is there to help and to say, well, here, let me get this information for you, or to take that basic information of age of onset. Do they have the DEE, diagnosis, and do they have the seizure frequency? Are they on excluded medications? A lot of those questions can be addressed, and then they'll be able to be triaged into an opportunity to be connected with the site. And, okay, hey, I'd like to ask more questions, and they can get connected to the site, and then be able to ask those questions to the physicians and see if this is right for them.
Cool. And when we think about phase 3, as far as, we were talking about phase 2, you had the option of doing it at home. So in phase 3, we're looking at the same options, right? So they can either go into the clinical site. So if they live close to a site and they want to physically go there, they can do that. Or they can do the at-home option, or they can do even a hybrid, right? So they can, if
That's
they
right.
live close enough and they want to go to a site, they can go to some of those site visits and then do the rest at home, correct?
That's correct. Yeah. Again, it's the story of the game here is flexibility. right? We want to be able to give them whatever fits their needs. And so it could be, like you said, they start out as a DCT or decentralized, where they go to their home. They say, well, I'd like to go in for this visit. We can arrange that so they can go in for the visit and say, okay, great, and then whatever fits their need and fits their life is what we're trying to do.
Yeah, which I appreciate it. As far as our families go, it's not a one size fits all. So being able to have these choices is really important and really just empowers the family too and the patient to figure out what fits best for us. So we do have a question coming out of Brazil. And so it looks like they're curious if the clinical trials it will be extended to Brazilian families with ADA. Is that something that you guys are looking at as a possibility?
So I'll let Brian Spar address that question.
Yes, well, certainly. So we're actively looking at all geographies right now. In Brazil, we just opened a trial for our ASO product for SCN2A gain of function. We know the long road it is to get a trial up and running in Brazil, but we've started that feasibility process with a couple of sites. The timing is probably a little less defined, and it's more likely tied to what comes next in terms of relutrigine in the clinic. So we're actively looking at expanding the breadth of DEEs that relutrigine might be amenable to. And if there's regions where we go with that study, but not in EMBOLD, we will include SCN8A in that trial as well. So,
okay.
I don't want to over promise anything, but it wouldn't be immediate, but it's on the horizon.
Would you say that it would be a very good idea for patients that are not US or in the EU at this point in time that's interested to go ahead and go to the nurse navigator and fill out those things so then you guys can see who all is interested in different areas?
I mean, there's certainly a value of adding names and of interest to help motivate us to say where these patients are. It is very hard to justify putting in the investment, both in people and money, of course, to a country or a site where there's very little patients or interest. So it's definitely worth, beating that drum. But I also encourage to consider the options of regardless where you live. And if we're not open in that country, we have come up with some creative solutions to get folks enrolled in the previous trial that we're not currently authorized to enroll in, but we find solutions. So if there's a patient that's either willing to travel or go to a different country in the short term, there's relocation programs that we have in place and ways to help families do that. So I wouldn't assume it's not an option until you ask the question.
Okay, I like that answer. Let's see. Another question we had was best case scenario. What would be the timeline of this medication being able to roll out to mass market and outside of clinical trials?
Great question.
Right, it's $1 million question,
well,
right?
So there's a couple of hard facts that when you pull up the study design, it's a 16-week trial plus 4 to 6 week screening and baseline period. So that's the actual term. Like I can't accelerate the calendar.
Right.
So the study has to be that, and that's in line with regulatory expectations and assumptions of what an appropriate trial looks like. Where we do have a way to accelerate is the pace of enrollment. So we can't read out a trial if we don't enroll as much as we can. So the faster we get to enrollment, the faster we get to data. So that aside, even if enrollment takes 6 months to 9 months, a year, you're looking at the back end of that of a 4-month trial plus a data readout and then a submission to the FDA or EMA. And then because this drug has orphan drug exclusivity, there's in theory, and we'd have to apply for priority review, but looking like 6 to 8 months for approval once we submit an NDA or new drug application. So probably looking at, I don't want to put a timeline, out there, but interval wise, probably a year from first patient in, we'd be in a position to at least have the discussion with the agency on our submission plan. Meaning when the final data is in, what needs to be kept in mind, and something we didn't get into and Brian didn't get into in the overview of drug development, we kind of ignored the whole technical world of toxicology, pharmacology, pharmacokinetics, and the chemistry and manufacturing that has to happen in parallel. So there's a lot of moving parts to get to an NDA and get to an approval. And there's certain things that we'll always continue to do to be creative and work with the agency to be flexible, because it's an orphan disease. But we're committed to keeping you posted on that timeline, and as we get any feedback from the agency, that's our priority, is to be transparent and share what we can.
Yeah, and I'll just say too, the impression that I've gotten from Praxis, especially in the last year, is the urgency. You all know the urgency. You want this to be as successful as we do. And so we've been really working hard together to push this as quickly as possible. And so that is something that I really appreciate from a patient advocacy role, is that, yeah, we're all wanting it to go as quickly as possible. And I don't, see any reason to why no feet are being drug on purpose. Let's say that. So good. Let's see here. We have another question. Where can we read more about the mechanism of action compared to other ASMs? Is there a specific science paper published on the molecular composition and its mechanism of action for this particular drug?
Yeah, we can get those. We can get those resources put together, and we can send them out after the meeting. But yeah, no,
that sounds great.
Yeah, great question.
Cool. Yep. Once I post, I can list everything that we've talked about too, and any links. I can post that in our own group and out in an email as well. Great question. And then we have one more. Is there any treatment for patients who might have absence seizures? Because right now it's geared towards more motor seizures, right?
Yeah, so that's so absence seizures are relatively difficult to treat in some ways. So we don't have anything within that's designed to treat the absence seizures yet. Keep you posted if any of that does change. This is typically like in our clinical trials, we're looking at the motor seizures, with regard to this drug. But yeah, I can keep you posted if anything would change in the future.
That sounds good. And I know sometimes, and I'm not familiar with the absence seizures as motor seizures, but I know once it gets to a point where doctors can prescribe, sometimes they can prescribe off-label to essentially see, if that would be helpful for those particular types of seizures. Okay, let me go back through and make sure I didn't miss anything on the question answers. Let's go back through. Families, it looks like we don't have any right now. This is a great opportunity to ask any more questions that you might have. And while we're waiting for other questions to come in, and we've got a few minutes here. And so I would say, if I, as a family member, we've been talking about this for a while. And it sounds like if I'm interested, this is something that I think could possibly work for my child, or at least I just want to know more. Maybe you're not even sure if you wanted to jump in full force. first thing you need to do is definitely contact that nurse navigator. Just because you ask questions and just because you're trying to inform yourself doesn't mean that you are locked in, right? So I think families, if this is something that you're like, I kind of think like this might be something I'm interested in, start asking questions, talk to your doctor, talk to the nurse navigator, start talking to your family. And then also think, we talked a little bit about this at Deep Day too, but it is a time commitment, right? This isn't a super easy thing that you're going to jump into, and it's not going to impede your life a little bit, right? So even in home visits, think about, you're going to have people in your house. They might be there for, hours at a time while you're doing blood draws, things like that. So think about that and see if this is a good time for you to do that, because it is going to be a commitment there. But if this is something that you're kind of thinking about, and maybe it's even been in the back of your mind for a while, I would really encourage families to look, see if it's an option. Because I don't know if we touched on this. How many patients are you guys wanting ideally for the phase 3 total?
Yeah, so we're looking at 80 patients.
80.
It would be
80. Yep. Mm-hmm. Yep. And that's going to be between the 2A and the 8A community. But the quicker we can get to that 80 patients, the quicker we can get data readout, the quicker all of this can go. And so I think we've mentioned it, but if we haven't, you guys are enrolling right now, right? So if this is something that parents are like, okay, I'm ready, I've been, thinking about this for a while, and they ask all the questions, you guys are raring to go and enrolling currently.
Cool.
Absolutely. And if there's any, like I've had times where clinicians or physicians have called me and said one of my patients, and they don't identify the patient or any of that, has expressed interest in this trial. Can you tell me more about it? That's something that we do offer, is additional, education, training, discussions, with the clinicians, because sometimes they're not as aware of all the details. So if anybody runs into that issue, I do have materials for physicians that they can look at, and in ways they can connect to me and schedule time with me if they wanted to, quickly review things. And we just want to make it, again, as easy for everybody to get the information that they need. So just wanted to throw that out there as an additional support.
I'm really glad you said that, Brian. I think that's a really good point, because some of us live in very rural areas, and it's a neurologist that's treating a lot of different types of cases, and so they may not be as up to date on different clinical trials specifically for 8A. So that's a great point, and I'll make sure we get connected and get those resources that I can give to families as well, so they know where to go if that's one of the roadblocks they're hitting. So
yeah, definitely.
Okay, I think we've got another question here as we're going through. I'm going through it. I think I may have already answered it and it popped up again. So I think we are good. All right, well, we are pretty much at time at this point in time. Oh, nope, sorry, one more. Is a tonic seizure considered as motor? And if they participate in the trial, will they have access to the drug until it goes public? That is a very good question.
So with the access to the drug, so yes, they would be able to stay as long as the drug is safe, and there's no issues. They would be able to stay on the drug until commercially available. So yes, that would be correct. Yeah. And then with regards to the seizure type, I'll reference back to the protocol just to double check. But yes, I want to, and we can send out that answer to the team. I just don't have the protocol in front of me at the moment. I just want to double check before I commit to that.
Yeah, and those are definitely details that can be worked through once you get to the nurse navigator and talk through to those next steps. So another reason that we can push to the nurse navigator for some of those questions as well. That was a really good question. All right.
Take a quick moment also, Casey, to thank you for your support and partnership. It's been a long road, but the progress has been great to see, and your leadership here is really fantastic. Appreciate it.
Thank you, Brian. Thank you. Yeah, this was all started before I jumped on. So I got the easy part of jumping on once the foundation was already set. But it's been a pleasure to work with you guys as well. And this is just really exciting for our 8A community and the 2A community for sure. But as this really is the very first clinical trial phase 2 drug readout for 8A, it's very exciting. And even just being able to see this grow, Stella's almost 9. So for 9 years, I've been able to sit back and watch all of this grow. It's been really fun to see and very, very exciting. So. And before we head off, I do, want to mention we have some amazing sponsors that make this education series possible. So we have our platinum sponsor, Neurocrine Biosciences, and our gold sponsor, Praxis Precision Medicine. And throughout the month of October and into November, we will have some more sessions for our Family Researcher Roundtable. So stay tuned as those become available, and I'll make sure to get all that information out to you families. And another thank you again. I know, Brian, you talked about it too, but for those families that participated in phase 2 and those that are looking at phase 3, thank you so much. Our whole community thanks you, and we know how big of a deal this is. And you and your kiddos, you're our superheroes, and we really, really appreciate everything you've done to move this forward. All right. Well, until next time, I appreciate you all, and we will see you on the other side.